get that started. So I'm going to start and just welcome everybody to this month's CIHD virtual seminar series. I want to just share my screen for a minute to show you a couple of things. Let's see, share, and I will bring them up. I'm going to share that screen and then get you to my email piece over here. So the thing I want to share for people is our website is undergoing some current revisions and remodeling. So I hope you'll take a look at it. It's not completed yet, but the most important thing that I want to show you is down here, which is a new funding announcement. So the good news for the CIHD is we competitively renewed, and so we have new funding for the next 5 years, and we have a new P&F opportunity right here that you can see, which is for 3 P&F grants that'll be at $33,000 a piece, and the proposal due dates are August 10th, and the instructions here are on the very front page of the CIHD website so that you can scroll down and see what you need to do. If you have questions, you should reach out to Dennis Wingy or John Phillips, and you can also reach out to me if I can help you in any way. And we're highly encourage individuals. We want to foster and support diversity among our researchers. And so if there's more questions about that, Cindy Roy at the NIH, who's a program director, is happy to talk to anybody about that. So I highly recommend you take a look on our website and check out that funding possibility for new research. And let me stop sharing now. And then I'm going to turn the time over to Paul to introduce our speaker today. And I want to thank you very much for speaking today. Okay, Paul, it's yours.
Great. All right. Well, thanks, Diane. Looks like a nice, intimate group here. So it looks like about half my lab. So I almost want to say Shay needs no introduction, but I will introduce him nonetheless.
For the rest of us.
That's right. Of course, Shay grew up in Nigeria and did his undergraduate work there, and then came to the US to do a master's in biochemistry in Tennessee, and before matriculating to the PhD program at the University of Utah, where he joined my lab and has been working to sort of understand adaptive metabolism, organellar metabolism, as he'll tell you about today. And specifically, we'll tell you about his work over the last 3 to 4 years, unraveling how a very divergent homolog of acyl carrier protein in the mitochondrion decouples iron sulfur cluster biogenesis from fatty acid synthesis and in Plasmodium falciparum malaria parasites. So we look forward to a good talk. Before I turn it over to Shay, just say feel free to pose any questions in the chat, or given how intimate a crowd we are today, I think it would also be fine if you want to just turn your video on unmute and ask a question as well. And obviously, Shay will be happy to take questions at the end as well. So take it away, Shay.
Cool. Thank you, Paul, for the introduction. And I really want to thank the organizers of the opportunity to talk about my thesis work so far in the Sigala lab. So malaria still remains a global challenge, as shown here by these statistics. It still affects over 200 million people worldwide, and it causes over 400 deaths annually. So the reason for this is we don't really have broadly effective vaccines, and due to the increase, and also another reason is an increase in drug resistance. So severe malaria is caused by this parasite called Plasmodium falciparum, which has a very complex life cycle. I'll walk you through the life cycle here. So it starts with a mosquito bite. So the mosquito injects sporozoite into the human host, and the sporozoites travel straight to the liver, which is now called the liver stage. They spend roughly 2 weeks in the liver and matures and ruptures again to invade red blood cells as merozoites. So in the red blood cells, we call that the blood stage. So it undergoes a 48-hour life cycle, starting from the merozoite to ring, to trophozoite, to schizon, and that cycle continues in a 48-hour manner rather. So a portion of this parasite differentiate into gametocytes for the mosquito to pick up, and the sexual stage continues in the mosquito. So all clinical symptoms arise in the blood stage. And we can readily culture this stage in the lab. That's why we focus more on the blood stage parasites. So in the blood stage parasites, the parasite lives in a very hemoglobin-rich environment, and it digests or sequester roughly 80% of the hemozoin into an organelle called the food vacuole in the parasite. So as shown here, the hemoglobin enters the food vacuole and gets degraded into two components, heme and peptides. So, so the parasite have a mechanism of detoxifying this heme to make crystals called the hemozoin, which helps them which shields them away from the toxicity of heme. In fact, chloroquine, a known drug, blocks the conversion of heme to hemozoin, and artemisinin in the parasite, which is a potent drug, is enhanced by the activity of heme in parasites. So in addition, the parasite has two critical organelles, the mitochondrion and the apicoplast. Of course, almost everyone knows what the mitochondrion is, but the apicoplast is a relic chloroplast that was gotten by secondary endosymbiosis way back. So these organelles house validated drug targets. For instance, doxycycline, it blocks translation in the apicoplast. Whereas Maluron, if you've traveled to a malaria endemic region, I'm pretty sure you must have been advised to take some prophylaxis called, like, for instance, Maluron, which is a combination of atovaquone and proguanil. So these two drugs block components of the electron transport chain in the mitochondrion. But these drugs that target the apicoplast processes and mitochondrial processes actually have some limitations. For instance, doxycycline has a slow activity. And these other drugs, atovaquone, proguanil, and other drugs known to target the process in mitochondrion, the parasites have developed some kind of resistance to these drugs. So those are the limitations. So there's need to actually identify new critical pathways in the organelles. So the question is, what are these pathways? So as listed here, I'm not going to go through these pathways today, but this question is a complex question as exemplified by fatty acid biosynthesis targeted to the apicoplast. So initially, the fatty acid biosynthesis in the apicoplast was thought to be a good drug target because you could target FAB-I with FAB-I was thought to be a good drug target because you can target FAB-I with a drug called triclosan and parasites would die. But when genetic studies came up, they ablated many proteins and enzymes in this pathway in the apicoplast, and parasites survived, meaning fatty acid biosynthesis is not essential in the bloodstream parasite. It's essential for other processes in mosquito stage and liver stage, but not for bloodstream parasites. So, in general, it's known that the fatty acid biosynthesis type 2 is targeted to the mitochondrion, at least in yeast and mammalian cells. And fatty acid biosynthesis, which I'll call FAST2 for short, is known to use that to make octanoate a precursor of lipoate, and lipoate is also a cofactor of pyruvate dehydrogenase in the mitochondria. But in contrast, checking the plasmodium mitochondria, it has lost all components of fatty acid biosynthesis, but retains an acyl carrier protein, which is curious. So what is acyl carrier protein? Acyl carrier protein is a small scaffold protein that helps in elongating the acyl chain in fatty acid biosynthesis process. It has this 3-helical bundle and has a very strictly conserved serine at this position that gets modified by 4-phosphopantethine, which I would call either 4-pp or PPANT for short. And this 4-pp gets the acyl chain, and it helps with the elongation of that, of the growing acyl chain. So when this serine is present or in the canonical ACP, it denotes that FASTO is present, and which is mediated by that serine I just talked about. But in contrast, when we looked at our mitochondrial SP in Plasmodium, that serine has been replaced with a phenylalanine, as shown here by the sequence alignment here. So that's the invariant serine I talked about earlier, and that's Plasmodium mitochondrial SP, which has been replaced with a phenylalanine. So which is consistent with loss of fatty acid biosynthesis in the organelle of mitochondria in parasites. So recently, a genome-wide knockout study showed that in rodent malaria and also in human malaria, it suggests that this protein, the mitochondria ACP in parasites, is essential based on this genome-wide knockout study. The gene was refractory to disruption, which suggests that it's essential, but it wasn't directly tested. So we wondered what the essential function of this non-canonical acyl carrier protein is. As I said, it doesn't have that conserved serine, it has been replaced with phenylalanine. So my project is actually centered on understanding the essential function of this non-canonical acyl carrier protein, which I'll walk you through the entire process of this talk. So to answer the question, we came up with three major questions. To first understand if the mitochondria is essential for blood-cell parasites, and if they're essential, what essential fatty acid biosynthesis independent interaction do they do in the plasmodium, and what cellular dysfunctions accompany the loss of mitochondrial ACP when we knock it down. So recently, it was the mitochondrial ACP was annotated, but it hasn't been studied, like I said. So we took advantage of the known sequence of mitochondrial ACP, and we tagged that with an HA, and we wanted to ask, does it even get targeted to the right organelle as proposed? So we did that, as shown here. The mitochondria ACP tagged with HA co-localizes with HSP60, a mitochondrial resident protein, as shown by this image here, and the nuclear DAPI stain. So to answer the first question I highlighted earlier, that is this protein even essential for blood-cell parasites? We took advantage of a CRISPR-based tagging. So to understand the essentiality of this protein, we used this CRISPR-Cas9 to tag the genomic locus of mitochondrial ACP to encode a C-terminal HA tag and a system known as the Aptamer TET-R-dosing system, which enables conditional regulation of the gene for protein expression using a non-toxic anhydrous tetracycline for ATC, which I'll call ATC for short. So that in the presence of anhydrous tetracycline or ATC, it binds to the TET-R-dosy. So the TET-R-dosy no longer binds to the aptamer, and you have active translation. And when ATC is washed out, the TET-R-dosy now binds to the aptamer and sequesters the mRNA, the MACP mRNA for degradation. So minus ATC, there's translation, the translation is off. So plus ATC, there's active translation, and minus ATC, there's, is transition off. So we took advantage of that, and we performed something called the growth assay. So I told you earlier that parasite has a 48-hour life cycle. So we synchronized parasites to early stage called the ring stage, and we measured parasitemia, which is the percentage of red blood cells infected by parasites every single day. And as shown here, plus ATC, when we have active translation of the mitochondrial ACP, we see this stepwise growth of parasites. And by Western, we could see that the protein has been expressed compared to the loading control AF1 beta. Also, by blood smear, we also saw that parasites were very healthy, as shown here by the parasites in the smear. That's the red blood cell. That's a red blood cell infected with a trophozoite staged parasite and a schizon stage parasite. So this parasite actually looked very healthy. So when we washed out ATC, we saw parasite failed to expand after the third life cycle. So, and as shown here by this blood smear. it was very difficult to even identify any parasite. I think the only one I identified here was dead. So, and by Western, we saw that the protein could not be detected. There was diminished protein level by Western. So, moving on to the next question, what the essential fatty acid biosynthesis independent interactions of mitochondria ACP are? So we asked a broad question: what other processes do we know that ACP even mediate, or other proteins that ACP interact with other than fatty acid biosynthesis in other organisms? So Angara in 2015 showed, in a review, showed that over the past decade, ACP is now known to interact with so many proteins called the LYR protein. It's a family of proteins called LYR proteins. And these proteins have they are small proteins and have this three helical structure. And as shown here, it interacts with many proteins, and these proteins are known to be assembly factors of the electron transport chain. So we wondered if these proteins are even conserved in parasites. So we took the amino acid sequence of each of these proteins and did a blast search to identify which of this protein is conserved in parasites. So to our surprise, IS11, which is shown here, that interacts with ACP and NFS1, is only homolog, LYR protein homolog, retained or conserved in Plasmodium. So, and that's a sequence alignment. At least showing the end terminal that our Plasmodium IS11 is an LYR protein, always found at the end terminal region of the protein. So it has a 30% identity to human IS11. And that's the process of making ions for clusters. So IS11 is an effector protein that binds to NFS1 to stabilize NFS1 to make ions for cluster. A collaborator at Johns Hopkins actually showed that these two proteins, NFS1 and IS11, get targeted to the right organelle. So to our knowledge, this has been the only extent that the ions for cluster has been studied in parasites. IS11 gets targeted to the right organelle as shown by colocalizing with mitotracker, and also NFS1 when target GFP colocalizes with mitotracker. Also, a colleague here in Utah, which I'm sure most people know on this group, Jared Roder and Dennis Wingy, showed that ACP in yeast and human interact with IS11 of the IS11 NFS1 complex via the prosthetic group I showed you earlier. So that if you mutate that serine and you don't have that prosthetic group, this interaction is ablated. And this entire complex makes ions for cluster.
Sorry, can I interrupt? This is Diane. Did you say they don't have an NFS1 though?
Sorry, if what has NFS1?
Your parasite.
It does have NFS1, but I'll get in a second. Yeah.
Okay, thanks. I misunderstood that. Thank you.
Yeah, sorry. So also, it was shown that the interaction between ACP and IS11, at least in human and yeast, happens via a sequence. Like I told you earlier, the LYR motif sequence. So leucine, tyrosine, and arginine. And if you mutate any of these residues, or if you mutate the LYR residues, you no longer have interaction with ACP. So the tyrosine and arginine interact with these charged residues here to mediate that interaction at that interface. So we wondered if our mitochondria ACP parasites interact with IS11 NFS1 complex in parasite. To answer Diane's question, yes, the Plasmodium has NFS1 IS11. So just to answer Diane's question. So what we did, we tagged the mitochondria ACP with HA, and we performed a non-biased IP mass spec study. So we did an immunoprecipitation to pull down the HA tagged ACP in parasites, and we send that for mass spec. So in all our mass spec data, we saw that NFS1 was highly enriched in almost all our data as compared to our control, the Picoplast ACP. And here I'm just showing you the bait protein. So the mitochondrial SP and apical plus ACP, they were both enriched in all experiments. So to test directly if ACP interacts with IS11, we transfected the ACP-HA line with a GFP tagged IS11, and we performed a reciprocal pull down, reciprocal immunoprecipitation to ask, do these proteins actually interact in parasite? So walking you through this block, when we did an anti-HAIP, so pulling down ACP, we saw signal for IS11. And when we did the reverse, pulling down GFP, so pulling down IS11, we saw signal for ACP. And when we did ACP alone, there was no signal for GFP. And as a control, we took advantage of the cytochrome C1, which is also a mitochondrial targeted protein, tagged with GFP, and we did the reciprocal pull down. So there was no interaction between both proteins. And also, to further confirm that, we did a reciprocal IP using the HSP60 that is mitochondrial resident, and we did reciprocal pull downs we've done. There were no interaction between those two proteins. So we came up with this conclusion that ACP interacts specifically with IS11 in parasites, despite the lack of the prosthetic group that was shown previously to be essential for interaction between ACP and IS11. So that was all done in parasites. So we turned to bacteria studies to actually ask, can we express these two proteins recombinantly in bacteria and see interaction in bacteria? So our ACP in bacteria was, so we expressed an HA tagged version of the mitochondrial in bacteria, and also a HIS tagged version of the IS11, and we co-express that in bacteria. And we asked by reciprocal pull down also, can we do this interaction? So as shown here, when we pull down ACP in bacteria, we saw interaction, we saw signal for IS11. And when we did the same for IS11, pulling down through nickel NTA isolation, we also saw signal for mitochondrial ACP. So I told you like some minutes ago that the interaction between ACP and IS11 occur via this LYR motif. So we wanted to know if we can mutate these residues and test what happens to the interaction. Do these mutations ablate the interaction with ACP? So we did that. We mutated tyrosine and arginine at this interface, and we saw little to no interaction signal for ACP when we pulled down IS11. And when we pulled down ACP, there was no signal for IS11. So, and also a colleague in the lab, Jaime Sepulveda, pulled down purified IS11 from the same bacteria and showed that ACP was, saw one-to-one complexation between ACP and IS11 when he did size exclusion chromatography. We concluded here that ACP binds directly to the IS11 and NF complex via the LYR sequence motif. So other than the LYR hallmark, there are other residues called lysine 44 and arginine 6 that have been showed structurally to mediate the interaction between IS11 and ACP, at least in human system. So lysine 44 and arginine 6 interact with the phosphate oxygen of the prosthetic group. The prosthetic group, the phosphopanthine. And a group in Pfizer showed that if you mutate this arginine to alanine, that mutation ablates the interaction between ACP and IS11. So like I've told you, that our parasite mitochondrial ACP lacks that conserved serine that would enable this modification. Imagine positioning these two residues, lysine 44 and arginine 6, close to that residue. We think the interaction between IS11 and ACP will be very, very unfavorable based on this type of interaction. So when we looked into the Plasmodium IS11, we actually saw that these two residues have now been replaced with more hydrophobic residues, cysteine at position 44 and isoleucine at position 6. So we think these mutations or this replacement have co-evolved with ACP to compensate for the loss of serine at this position. So like I told you like a minute ago, that ACP IS11 interaction, occurs via this prosthetic group and via this isolated prosthetic group, anagenine 6. So when we sent our mitochondrial ACP sequence to a collaborator at the Baker Lab at the University of Washington, and he showed that the phenylalanine is positioned in a more hydrophobic environment, as shown here by this in silico structure. So we wanted to test directly, do we see any influence of this phenylalanine? Does it ablate interaction, or what does it do in parasites if we mutate that to alanine? So we turned to bacteria again. We mutate this residue to alanine and asked by reciprocal by pull down what happens to the interaction. So as shown here, when we mutated that residue to alanine, we no longer see interaction with ACP, showing that this residue contributes to some interaction or dye interface, or the kind of interaction it does at that interface. And we did a complementary assay in parasites. So in our knockdown line, we transfected the knockdown line with either Y-type episomal ACP or the mutated episomal ACP, and asked by complementary assay and growth assay, like I explained earlier, what happens to parasites. So the Y-type ACP completely rescues parasite growth compared to when we mutate that residue to alanine with some modest rescue. So we came up with the conclusion that this phenylalanine contributes to the binding interface and it does something important for interaction between ACP and IS11. So far, going through the questions I highlighted earlier, is mitochondrial ACP essential for blood parasite? I think we now know that the answer is yes, it does. It's really essential for parasites. And answering the second question, we now know that ACP interacts with the IS11 NFS1 complex, despite the lack of phosphopantethine in this position. So moving on to the last question, to understand what the cellular dysfunctions are that accompany loss or knockdown of mitochondrial ACP, we know that the ion spore cluster pathway, also known as the ISA pathway in Plasmodium, supports ion spore cluster proteins within the mitochondrion. So risky is a component of the electron transport chain complex 3, fumarate hydratase, ferredoxin, which is also involved in ion spore cluster biogenesis itself, aconitase and succinate dehydrogenase. They are both components of the TCA cycle. So it's also known that the mitochondrial ion spore cluster pathway supports processes in the cytoplasm and the nucleus, ion spore cluster processes in these two compartments, cytoplasm and nucleus. John Van Vranken, a former grad student in the Rotter Lab, showed in his 2016 Eli paper that if you ablate ACP, you lose NFS1. It impacts NFS1. So we wanted to test this directly in parasites. Do we see any impact of ACP knockdown on NFS1 in parasites? So we did that. Upon washing out of ATC, which means ACP is knocked down, we saw no signal for NFS1 compared to plus ATC and the loading control EF1 alpha. And so it's known that loss of NFS1 should impact ion spore cluster biogenesis itself and the client proteins that need ion spore cluster. So the question is, what are the ions for cluster proteins? Like I said earlier, TCA cycle, aconitase and succinate dehydrogenase are involved in the TCA cycle. So these two proteins, and TCA cycle is not essential. It's been shown that TCA cycle is not essential for cluster parasites. Ferredoxin is also involved in ions for cluster biogenesis itself. And fumarate hydratase has some conundrum in its essentiality, which I might talk about briefly at the end of this talk. And risky, which is a component of complex theory, is known to be essential for processes in the mitochondrial and electron transport chain. So we took advantage of that and asked what happens to risky upon knockdown of ACP in parasites. So this is just structure. That's where risky is positioned. So complex theory, just reiterating what I just said earlier. And we, when we washed out ATC, well, sorry, what we did, let me backtrack a little bit. We transfected plasmid expressing risky HA into the knockdown line we had and asked if we wash out ATC, what happens to risky? As shown by this plot, upon knockdown of ACP, when we wash out ATC, we could no longer detect risky HA compared to the loading control EF1 beta. So loss of risky is expected to inhibit the electron transport chain. It should impact the electron transport chain. So we plan to test this in two ways, which I'll walk you through in a second. The electron transport chain in glossy parasite has two essential functions. One is to pump protons, and also to recycle ubiquinol for processes for pyrimidine biosynthesis and other downstream processes. So, and also, this electron transport chain has a secondary proton pumping pathway, so that if you block the first, if you block cytochrome B, a component of electron transport chain complex theory with atovaquone, you don't depolarize the mitochondria. The mitochondria, as shown by this image here, showing a punctate signal for mitochondria using the mitotracker. So the reason for that is that this pathway becomes essential to maintain the membrane potential in parasites. Only when you block both pathways do you see the collapse of the membrane potential, as shown here by mitotracker. So it's now dispersed. The mitotracker is now dispersed within the mitochondrion and the cytoplasm, and you see that dispersed signal when you block with both drugs. So we want that upon knockdown of ACP, which should mimic atovaquone treatment, and in synergy treating with proguanil, do we see the same effect we just saw? So we did that. Plus ATC alone, we saw punctate signal for mitotracker. Plus ATC and proguanil, at least at the concentration, non-lethal concentration we use, we also saw punctate signal for the mitotracker. And also just knocking down ACP minus ATC, we also saw punctate signal for the mitotracker red, which showed that the membrane potential were intact. So only when we knocked down ACP and treated with proguanil, we only saw, that's when we saw the dispersed signal for mitotracker, showing that the electron, showing that they both work synergistically to impact the membrane potential in parasites. So that's the first way to test if loss of risky impacts the electron transport chain. So the other way to do that, like I just said earlier, ubiquinol recycling is the sole essential function of the electron transport chain, at least in glossy parasite. So if you block cytochrome B in complex theory with atovaquone, you can rescue that lethal phenotype with decol-ubiquinone, which is a soluble analog of ubiquinol, as shown here by this plot. Again, we're measuring parasitemia, and that was done each day. So if you treat with atovaquone is lethal to parasites, but you can rescue that phenotype when you supplement with decol-ubiquinone. So we want that in parasite, like I showed you earlier. can we knock down mitochondrial ACP and also rescue with decubiquinol? So like I showed you earlier, if you knock down ACP, the rescue is done, which mimic atopicon treatment. So we did that experiment. We knocked down ACP. So minus ATC is ACP knockdown. As you can see that parasite failed to grow past the second cycle, and plus ATC parasites were doing very fine. So if you knock down ACP minus ATC, you can rescue that phenotype up to the third cycle, which I would explain in a second. So at this point, we feel we concluded that the electron transport chain dysfunction is a dominant cause, immediate cause of parasite death, because we can rescue that phenotype with decubiquinol up to the third cycle. So long term, we saw here that after the third cycle, decubiquinol fails to rescue parasites growth, as shown here. After day 5, parasites started dying. And compared to atopicon treatment, like I showed you earlier, atopicon rescues parasite indefinitely compared to decubiquinol in minus ATC. So here we now concluded that an essential pathway beyond the electron transport chain becomes limiting long term. So we think that is the ion spore cluster by processes outside the organelle in the cytoplasm of the nucleus, or it could also be the fumarate hydratase I talked about briefly earlier. So these are the proteins I showed you. So that's the IC pathway, which supports processes outside the mitochondrion, cytoplasm, and nucleus, and risky, which it gets impacted upon knockdown of ACP. So these two processes are not essential for bloodstream parasite, and ferredoxin is also part of the ion spore cluster biogenesis itself. So we feel the phenotype we see here for why DQ doesn't rescue past the third cycle could either be due to this fumarate hydratase that's been shown to be essential for bloodstream parasite, or processes outside the organelle, probably in the cytoplasm or nucleus. So in summary, I've shown you today that ACP interacts with IS11 despite the lack of that prosthetic group, which is known to mediate interaction between ACP and IS11. And this complex, ACP, IS11, NFS1 complex in parasites, makes ion supercluster to support proteins listed here, like I told you earlier. So that if you knock down ACP, you impact NFS1 and you see degradation of NFS1. Also, the rescue component of complex 3 doesn't get ion supercluster, and that leads to loss of rescue. So there are some evidence that this complex also supplies some supports the ion supercluster processes in the cytoplasm and nucleus. That's why there's a question mark. It's not really certain, but we think there are some processes from this complex that support the process in the cytoplasm and nucleus. So in conclusion, parasites retains an essential mitochondrial ACP despite the loss of the prosthetic group I showed you earlier, which is a new in this field. That without the mitochondria fatty acid biosynthesis, parasite is still essential in mitochondria. And ACP binds to the IS11 NFS1 complex to mediate ion supercluster biogenesis, like I've talked about for the past 30 or 40 minutes. And we now know that the electron transport chain, which at least by electron transport chain dysfunction, at least by the loss of risky, is the immediate cause of parasite death when we knock down ACP. And the knockdown of ACP causes parasite death on a slower time scale, which we think is due to the dysfunction beyond the electron transport chain, like I explained like 2 minutes ago, which we think might be the cytosolic ion supercluster pathway, or also the fumarate hydratase, which is thought to be essential for bloodstream parasites. So the significance of this study is mitochondrial ACP in parasite has now evolved to decouple iron spore cluster biogenesis from fatty acid biogenesis and biosynthesis type 2 in parasite. The divergent interaction between ACP and IS11 provides new paradigm for association between ACP and IS11 without the isolated PPANs, prosthetic rule or 4PP. And we think this interaction is novel and can be leveraged leveraged upon for new drug targets in the mitochondrion. So mitochondrial SP retention without FAST2 actually highlights the fundamental role of iron spore cluster biogenesis in eukaryotes. And loss of fatty acid biosynthesis and retention of this divergent ACP that I've shared with you, we think there are shared metabolic features of other epicoplex proteins that affect human, for instance, the toxoplasma and babesia that are known to infect humans. So with that, I would like to thank the Sigala lab for the contributions, and also our Dennis Wingy, Jerry Roda, and Sean Priggy, who have been super supportive intellectually to this project, and our collaborators at UCLA, Yazi, Jamie, and James Wash-Lego. They've really helped with the IPM aspects of this. And Humbean Park and in University of Washington in the Baker Lab, who did the in silico modeling for ACP and IS11, and also our funding sources. Thank you. I'll take any questions.
Great, thanks, Shay. Happy to open it up if anyone has questions they would like to pose, Shay.
I have a question. It's Diane Ward again. Do you know how iron gets in or iron sulfur clusters get out in Plasmodium?
Well, we don't know how iron gets in. I think that's still something people are still trying to figure out in Plasmodium, because I think for human it has a frataxin, but parasites don't have that. So it's still something we're still trying to figure out how iron gets in.
Do they have a mitoferrin?
Mitoferrin?
That's the iron transporter in yeast and mammals. There's two of them.
They have DMT1. I don't know, but I don't know if they have any homolog for mitoferrin.
Okay.
There's a DMT1, yeah.
Yeah, we should look at that, Diane. It's a good question. In general, pretty much all of the conserved iron storage and transport proteins, there are few, if any, homologs in Plasmodium, which presumably they're present and they may be sufficiently divergent that we just can't identify them. It's unclear. Right on the iron sulfur side, the model has been that this ATM1, transports some sulfur intermediate to the cytoplasm, at least based on studies in yeast. And there is one homolog of that. It's never been localized. It's not clear it's mitochondrial, and it's not essential for the blood stages. So there's clearly a lot of work to do to understand some very basic aspects of iron metabolism.
Are there other known metal importers? I mean, to get any other metal into the mitochondria? Have any metal importers been identified?
I think other than the DMT1, I don't know if there's any other one.
So the DMT is on the plasma membrane, right? Or is it found on the mitochondria?
So I think that's found on, I don't know. So a grad student in the lab is actually working on that, but I think it should be found on the mitochondria, if I'm not mistaken.
Well, so that's hasn't been directly, there's one DMT1 divalent metal transporter, one homolog. It hasn't been directly studied, has been rumored to go to the food vacuole membrane, but has not been directly localized. There's also another so-called vacuolar iron transporter that has been studied in more depth, but that appears to go to the ER and has been suggested to mediate sort of an iron overload protection mechanism by shunting iron into the ER. There really hasn't been anything formally studied in the context of the mitochondrion, but I would say this is an aspect of parasite metabolism that has been largely neglected. So I think there are a lot of ripe questions such as that, Diane, which are wide open to study.
Yeah, sorry, I keep asking.
No, these are all good questions.
Why would you store iron in the ER versus in the vacuole, like everything else does?
Well, there probably is iron
Yeah.
generated, right, in the vacuole. Yeah, good question. That the localization to the ER for that VIT homolog has never made a lot of sense for me. It's been studied a bit more recently in toxoplasma and suggested to maybe that ER localization may not be entirely accurate.
Yeah.
Yeah. So there's some fundamental questions that don't quite add up.
Yeah. Okay, cool. I could go on, but I'll stop.
Thanks for the question, and yep, that's some good things there. Any other questions for Shay? Again, I realize this is predominantly my lab that's still on here, but anybody should feel free to fire away.
I'm going to ask another.
Go for it.
I should know this, Paul, but it could be drug-induced questions here. But does Plasmodium have ferritin? They have any other kind of iron storage capability?
You want to take a crack at that, Shay?
I think the ferritin. Do they have ferritin?
Yeah, they basically have no obvious homolog of ferritin. There's if you blast the Plasmodium genome with, human ferritin or whatever, there's nothing that comes up there, which again, if they have an iron storage protein, maybe they utilize something else, or maybe the sequence is so divergent
Right.
it's not readily identified. I mean, there is, some thought of how do they access iron in general, right? I mean, it is known that exogenous iron is absolutely essential for parasite viability. How they acquire that is not entirely clear. There's reasonably good evidence that it's mediated by uptake through the digestive vacuole, which either could reflect a trace amount of non-enzymatic heme degradation in that vacuole, or they probably during hemoglobin uptake take up a small portion of human ferritin in the red cell cytoplasm that may have iron associated, which might get released in the vacuole. But how these get stored and trafficked are wide open questions.
Okay. Thank you. Thanks for the great talk.
Well, thank you.
Yeah, thanks everybody for chiming in today and listening to Shay present his thesis work.
Yeah. Okay. I will